Hello, and welcome to Conservation Skills in 10 Minutes or Less. This series of short, skill-based videos is brought to you by the U.S. Fish and Wildlife Service's National Conservation Training Center in Shepherdstown, West Virginia. If you have a couple of minutes, pull up a chair and pick up a new conservation skill or maybe refresh an old one on topics ranging from fish culture to bird identification to stream restoration. Enjoy.
As I mentioned before, with algae cultures, you have to step them up in size. The first step that we talked about was the slant culture. That's the smallest of the stepwise. If you went right from a slant to, say, a huge container, say a 1,000-liter container with that small inoculum, there wouldn't be enough algae in there for it to get started properly, and any light that's coming in, that UV light, is going to break down the cell walls. So it's important to just step it up little bits at a time, allow it to get dense in that next culture, move it up to a little bit bigger, allow it to get dense, move it up a little bit bigger, allow it to get dense. So this is the second step. This is flask culture. And you can see on this flask here, we have the autoclave tape around the top and covered with aluminum foil. So we know that this flask, the inside of this flask, is completely sterile. So we're good to go there. So what I'm going to do is transfer the culture from the slants that we just used now to this second step. So obviously, as I mentioned before with the slant culture, all of this needs to be done under the hood in a sterile environment. I'm only doing it out on this table for demonstration purposes for the camera. So always keep that in mind. So first thing you need to do is take obviously the aluminum foil off. And you can see in the top there is a foam stopper. This stopper prevents air from getting into the culture and contaminating the culture. Any air anywhere has algal cells, bacteria, fungal cells floating around in the air. And you have to keep that from getting into your culture if you want to maintain monocultures. So you put this foam stopper in first before the autoclaving process so that the flask and the stopper are sterile. Then, in addition to the media, which we're going to add, obviously algae need light to grow, nutrients to grow, and they also need carbon dioxide to grow. So we need to have some way to add air to this system. And that's what this 1 milliliter pipette is for. We've just turned it basically into a bubbling tube. So again, anything that's going to be in contact with the algae, in other words, anything that's beyond the stopper, you don't want to touch. So you take this out, not touching it to anything, and inserting it into the foam stopper. In this case, we already have a hole in the top of the stopper, which aids in getting the pipette through the hole. Okay, so now you have your flask, your culture container, which in this case is a flask, your foam stopper to prevent air from getting in, and your bubbling tube. You'll notice too that the bubbling tube, or in this case a pipette, has a little piece of cotton in the end. That also provides some filter for the air that's going to be coming in. So the next thing we need is the growth media. Just like any other plant, it has to have fertilizer of some kind to grow. This is pre-made up and has been sterilized. You see it says sterile water. You sterilize the water first in an autoclave, and then you add your media to it. So I need to be able to get this stopper out without touching anything. What I like to do, sometimes you can grab the top of it and pull. If it won't come out, you can take this tube and kind of push it to one side and it lifts right out. So you want to go ahead and loosen the cap on your media. Take the cap off, lift the stopper out, and you want to add about 300 milliliters of media to a 500 milliliter flask. It doesn't have to be exactly 300. and then put the stopper back in. So now what we have is our culture flask, our growth media, our foam stopper to prevent air from coming in, and our bubbling tube. The last thing that we need is our inoculum, or the algae. And that process is very much the same as adding algae, going from slant to slant, and except in this case you're going from slant to flask culture. So again, you need your sterile loop just like we used before. In this case I'm going to use the large loop because I want to get a large inoculum in here. And just as I mentioned before, if there's not enough algae in here and it's not dense enough, the UV light coming from your light source can break down the cells and cause your culture to crash. So we want to get a nice strong inoculum in there. The next step is to transfer the inoculum to the flask, but I can't stress enough, it's important before you do that, you want to put a label on this flask. And the label, we like to use colored tape, and you can even use a particular color for a particular algae species if you like to keep things straight. But once this inoculum goes in here, a lot of these species are green. You're not going to have any idea what species you have. So it's really important before you put the inoculum in to put a label on this that tells you the species that's growing in there and also the date at which you place the inoculum in the flask, because not only does that identify what species you have, but you also are going to be on a schedule where you're going to back these things up just like the slants every couple weeks or 3 weeks. So you need to know exactly what date this culture was started so you know when to back it up. So since I'm going to use the big looped end, I want to open it from the small loop end. Again, not touching the loop to anything. Same as before in the pinky to get the inoculum out of the tube. You can see in this case I'm going to get quite a bit larger inoculum. Again, pull the stopper out and just kind of shake it back and forth until you see that those algae cells come off of that loop, and put the stopper back. So now that we have the inoculum in the flask, we have our media, our bubbling tube. We just need to put it on the aeration at this point. Remember, it has to have CO2 to grow. So we just have this little aeration apparatus. You can use whatever aeration apparatus that you want. In this case, this particular diameter of tubing fits right on the end of these 1 milliliter pipettes. And then these little clamps, you just open the clamp and it starts bubbling. You also notice that our aeration apparatus has this inline filter. Because we're just pulling in regular air from the outside, I mentioned in previous videos that there's algae, fungus, bacteria floating around in the air. So it's important to have this air filter in line because that's another source of contamination to your culture. So we keep our slant cultures and also our flask cultures in this temperature controlled incubator. The reason we do that, we can keep the temperature, you can see we keep it at about 18 degrees C. That just allows the cultures to grow slowly and stay nice and healthy, and they don't outgrow their container. It also provides a very nice light source. So it's just a very controlled environment. And anytime you have these early cultures before you step it up even further, it's nice to have a temperature controlled environment for them to grow. So early on in this stage, we recommend indirect light for the cultures. So you can take, this is just a piece of plywood, you can take a piece of cardboard, whatever it is, and just set it in front of the cultures temporarily so that the light coming from the front of the incubator is not directly on the algae. It's actually bouncing off the back and hitting the algae. And then when it grows for maybe 3 or 4 days and starts to get some color to it, you can take this piece back off and you're good to go.
Thanks again for joining us for Conservation Skills in 10 minutes or less. If you enjoyed this video, please give us a like or hit the subscribe button, share this video with a friend, or even check out one of the many other skill-based videos we have in this series. Have a great day, and always remember, the beautiful thing about learning is that no one can take it away from you.